and we can use them to prove our facts. So first thing we're talking about is the serological tests, and how these serological tests will help us to understand who is related to whom. So as we can look, this serological test, as the name suggests, is a test by using antibody and antigen. So using antibody and antiserum, which has been produced by a cell when the bacteria enters into a body. So if we put bacteria inside the cell of a horse and produce antibody against that, so it will produce some of the antibodies. Now we have those antibodies. Now what we can do, we can use those antibodies. For example, in this case, in this picture we can see, if we use this antibody and use another antigen producing bacteria. So suppose this is our known antibody. So our known antibodies, so this orange color antibodies is our known antibodies, and we know now we have a bacteria. These known antibodies are being produced by a bacteria A. So bacteria A produces this known antibody. Now suppose we have a new unknown bacteria. Now we have an unknown bacteria. So this unknown bacteria, which is bacteria UB, I say unknown bacteria. Then this unknown bacteria produces an antigen. Now this antigen is unknown to us. Now what we can do, we use this antigen along with that bacteria which is producing that antigen inside a serum. Now use this known antibody to act against this antigen. If this antibody will act against the antigen and coagulate that antigen, and it will coagulate the antigen, then we can say that that this bacteria which we are talking about is very much similar with the bacteria we know. So this UB will be similar to A in many aspects. How? Because UB is producing the antigen and the antibody is produced by A. So this antibody has an ability to be attached with to be interact with the antigen produced by the unknown bacteria. That means they have some similarities in the production of antigen. They have some similarities among themselves. That's why they can attach or agglutinate all these things together. So we can almost, we cannot say in detail, but we can say, yeah, they are in a relationship. And what is the advantage of serological test is that they are highly specific. So we can tell at least by looking at this that yes, this is a fact, because otherwise this antigen antibody will not interact, because antigen antibody interact only when everything matches together, right? Okay. Now we can also use this kind of test using fluorochrome. So you attach fluorescent we have fluorescent dyes with the antibody. The antibody will get fluoresced. Then we can cell mixture, and we can make a suspension of cells and we add those antibody. If the bacterial antigen is recognized by the antibody, then the bacterial cells will be coagulated, and we can look at the fluorescence amount and how much bacterial cell will be coagulated is being coagulated. By looking at this, we can say yes, this bacteria A and B are linked with each other or related with each other by using this kind of serological test. So this is the type of test that will help us to understand specifically who is related to whom. But this test actually helps us to understand the differences between species only, because if we go from look at the previous picture, if we go from species, there are minor differences, and these antibody antigen interaction tests are very sensitive, as we know. So they are only able to tell us about the differences between species. But if we think the difference between genus, if we try to maintain the difference, if we try to know the difference between two genus, then this serological test will fail, because one genus of bacteria produce some of the antibodies. Those antibodies will not have any kind of specificity with another antigen with another genus. So the intergenus interaction between this antibody antigen will not be accomplished. Okay, so that's why this is the drawback of this serological test, is that we can only use this kind of test to distinguish the type of species what we have, or in very few extents the genus, but not more than that. Okay. So another advantage is does not usually require the organisms to be isolated into pure culture, because we can use it, we can make just a culture, make a suspension, then we can use it, because it's specified, then the other debris which are present will not interfere with the interaction of antigen antibody. Okay, so it can be used to identify between some of the bacteria which cannot be grown on the medium. So non-cultured bacteria, in case of non-cultured bacteria, we can use this kind of tests. But the drawback is we can only do at the level